Well, hello and welcome to our Hearing Health Foundation research webinar. Please do enable the Zoom captions by clicking the CC button in the toolbar at the bottom of your screen. These captions can be moved anywhere on your screen. Now, if you need any other assistance using Zoom, follow the link to the technical guide shared in the chat. Also, this event is being recorded and will be posted with captions as well. Now, by way of introduction, my name is Dr. Anil Lawani. I'm a professor and vice chairman for research in the Department of Otolaryngology, head and neck surgery, or ear, nose, and throat, as well as the associate dean for student research at Columbia University, Vagelos College of Physicians and Surgeons in New York. I'm also a board member at Hearing Health Foundation and the head of the Hearing Health Foundation's Council of Scientific Trustees, which oversees a program called the Emerging Research Grants Program, or affectionately known as ERG. Well, ERG is a competitive program that funds researchers conducting cutting-edge hearing and balance research. Now, these grants have supported many leaders in our field to become very successful scientists, including Professor Heller, our speaker today. And Professor Heller, we take all credit for your success in your entire career based on that funding. Now, let's get to the topic of the webinar today. We're still learning a lot from birds about sensory hair cell regeneration. Now, the discovery that birds can regenerate their auditory hair cells and naturally restore their hearing sparked an ongoing investigation into how that happens, how this remarkable ability occurs at the molecular level, actually leading to the creation of Hearing Health Foundation's Hearing Restoration Project. Now, in this research webinar, Hearing Restoration Project member Stefan Heller will discuss how innovative technologies bring us closer to the understanding of the molecular signals that regulate the production of new hair cells in the inner ear of chickens. Now, these insights open up new avenues for research that can be tested in mammals, us, which lack the ability to regenerate hair cells. Dr. Heller, Professor Heller, is a professor of otolaryngology head and neck surgery at Stanford University and the Edward C. and Amy H. Sewall Professor at the Stanford School of Medicine. He's also a faculty member at their Institute for Stem Cell Biology and Regenerative Medicine. And as I noted earlier, he was a proud recipient of the 2001 to 2002 ERG Scientist Award. Well, Hearing Health Foundation's ERG program that provides seed money to scientists who are starting out in their field of research is only possible through the generosity of supporters like you. Now, if you'd like to support our work on hearing loss, tinnitus, and related conditions, you can do so today at HHF, Hearing Health Foundation, hhf.org/donate. Now, without further ado, we're going to move on to Dr. Heller's presentation. Please do ask your questions through the Q&A box linked at the bottom of your screen that we'll try to answer following the presentation. Professor Heller.
Thank you, Anil, for this nice introduction. And thank you, Hearing Health Foundation, for the ongoing support that truly helped when I started out to start my laboratory back in 2000, back in Boston. And I'm now in California and still going strongly. So as probably all of you know, hearing loss has many causes, and aging, genetic mutations in certain genes, noise exposure, drugs. There's also a very interesting kind of phenomenon that some of us have really sensitive ears, whereas others have iron ears, which is an interesting topic by itself. And I will not talk about this today. I just want to note it. With all of these different causes, there's a common result. And this is hair cell loss. Hair cell loss can be primary, or it could also be secondary. But when hair cells are lost, you cannot hear anymore. And there are two examples shown here on the right. On the top is an example for the loss of outer hair cells, which are the more sensitive type of hair cells. And these hair cells are responsible for amplifying sound. And in instances like this, a hearing aid might provide some help because it replaces the amplification that is lost due to the death of these outer hair cells. When the inner hair cells are gone, which is shown on the bottom, there is no way that the ear can hear naturally anymore. And a cochlear implant is the only solution that we have for these patients. This is summarizing it. No more hair cells means hearing loss. And I always imagine it having a stage where there's a performance and you steal the microphones. There's no more that you can hear. So getting these microphones back, plugging them back into the cables that are still there would actually be quite an effort to restore hearing. And this is what I hope to be able to do in the long run. And the reason we are thinking that this is possible is if you look into species that are not mammalian, for example, salamanders. Salamanders, as many of you know, can regrow their tails. They can throw off their tails, or if you be as cruel and cut it off, they will start growing new tails. And what is interesting, part of the tail is an organ that's called a lateral line. And this lateral line organ in amphibians, as well as in fish, contains hair cells that have the same evolutionary background as the hair cells that we have in the inner ear. And these hair cells form in these newly regenerated tails. So this is an early indication that hair cell regeneration might be possible. Of course, the breakthrough came in 1988 by two back then competing research groups. And this discovery was made by Brenda Riles and Ed Rubel, as well as Jeff Corwin and Doug Kotange. And they found that in birds, like chickens and quails, new hair cells emerged after acoustic trauma. And the new hair cells are labeled here with these radio-labeled dots, these dark spots that are overlaying the hair cells and the supporting cells. This was the technology back in the 80s. But it was clear. It led to two publications in Science, and it caused a lot of excitement in the field. By the way, Doug Kotange was also an HHF grant recipient back then. I mentioned it led to a lot of excitement, and this was mainly because during this time, there was a group of molecules identified that were called neurotrophic factors. And neurotrophic factors provide trophic support, meaning they help neurons to survive. And this raised the question whether these neurotrophic factors might be applicable to treat neurodegenerative diseases like Lou Gehrig's disease, where neurons die, or Alzheimer, or certain diseases that end up with dementia. And as we all know, it's a long road. These neurotrophic factors prove to be useful for certain things, but not for everything. But the ear field was also playing a lot with neurotrophic factors. And this is when I entered the field. I also back then appreciated that developmental neurobiology received a renaissance where people studied how the brain is being built in an embryo. And for the inner ear, that meant people studied how the ear is being built, because knowing how the organ is being assembled provides information potentially for how to fix it. So knowing how it's being put together helps, because then you know, maybe I can fix it in a certain way. So this kept people busy through the 90s. The problem was that in comparison to other organ systems, the inner ear has a problem. And that problem is that the cell types that are of interest are really rare. There are only 3,200 hair cells in a mouse cochlea. And the supporting cells, which are the source for new hair cells, are equally rare. And when you compare this with, for example, the eye, the retina has several million photoreceptors. A human retina has 120 million photoreceptors. And I remember when I was a high school student, we had to dissect eyes that our teacher got from the butcher, and she had a bucket of eyeballs. And back then, I didn't really appreciate that, and it made me really feel uncomfortable dissecting these eyes. But over time, I appreciated learning biology, and actually, this is actually a good way of getting material back then. The eye field was way ahead of the ear field for quite a long time. A second major constraint was that tissue culture of inner ear cells is extremely challenging. Taking out tissue, dissecting it from a mouse, and then putting it into culture works in with embryonic tissue. Embryonic tissue survives and continues to develop, but once you take tissue out of an adult mouse, and this is the substrate that we work with, we want to instigate regeneration in an adult animal, the cells just die. They don't like it to be in the culture. In addition, human inner ear tissue cannot be assessed. I mean, we cannot just take ear tissue out of a human being without causing hearing loss. So this is a rather limited source for tissue for doing experiments. That slowed down research in our field for many years, until about 10 years ago, when two technologies emerged that my lab immediately jumped on because I like new technology. And one technology is the molecular profiling of individual cells. That means you can take a single cell and you can get a lot of information from the cell. The methods to analyze individual cells have excelled over the last 10 years. That means that the low cell number is no longer an issue. It's actually a virtue, because now we can oversample the ear. We can sample every single cell in the ear, whereas the people in the eye have to deal with 100 million cells. So this is a cost factor. They cannot do this cheaply. Plus, stem cell biology has led to the technology that allows us to grow ears in a dish. This is not perfectionized yet, but it's getting there. So we can generate inner ear cells in a culture dish. And these cells are happy. That means that they are not instantly dying when we do something with them. So we can now build on that. Our lab works with both of these technologies. I will mainly talk about technology number one today. What has inspired me to come back, because my lab was doing work on ion channels back in the days, and what inspired me to come back to looking into regeneration. This is one of the findings that actually was really inspiring. It's a small paper, but it shows that when you look at starlings that are being kept in a cage in an animal facility or in a zoo, and you measure the hearing of the really old birds that these birds are 8 to 13 years old in this circumstance, people notice that they do not suffer from age-related hearing loss. The natural life expectancy of a starling in the wild is 4, 5, 6 years. So these are really, really old animals, and they have perfect hearing. So combined with the knowledge that the inner ear regenerates, that provided some insight that the birds are potentially capable of maintaining hearing health throughout their life. And we wanted to know how this works, right? So this is an example, an illustration how the chicken ear looks when you take it out of the animal, shown here. It's not a curled structure like a snail, like the cochlea. The chicken ear is more like a tube. It's not curled up, but it's the same kind of principle. When you cut this tube, like you cut a slice of a salami, you get a picture that is shown on the right. And you see that the chicken inner ear has what we call tall hair cells, and it has short hair cells. and they are the functional equivalents of the inner hair cells and the outer hair cells of the mammalian cochlea. So there are some structural similarities between these two ears, and we think they can be used for comparative studies, right? This is how we see this ear. So this is a view through the microscope, and when you think about what I just showed you, the artist's illustration, this is real life. So the hair cells are shown in magenta here, and the supporting cells are not shown in their full glory because they're really they're cells that are really difficult to visualize. But what we can visualize are their nuclei. The nuclei is the place where all the DNA, the genetic information is stored in a cell, and we visualize their nuclei with an antibody that labels these supporting cells in this nice turquoise color. And you see all the other structures in the inner ear. So for the purpose of what I will be talking about today, I will focus mainly on the different supporting cells that are underlying the hair cells. So when we infuse an ototoxic drug into the chicken ear, we cause hair cell death. And that is shown on the bottom slide. This is one day after we infused an aminoglycoside called zizomycin, which is highly ototoxic, into the chicken ear. This is the left ear on the bottom, and on the upper you see the right ear. So we can do this unilaterally. We can deafen the chicken only on one side. And the control ear is perfectly fine. Same picture as I showed before. And on the bottom you see that the hair cells are rather, they're kaput. They are really destroyed. And what you see here is only the debris of the hair cells that have been expelled from the sensory epithelium. The epithelium has closed up, and on the bottom you see the supporting cells shown in green and in turquoise in this area here. You also see that it has flattened quite a bit. This is the same picture 5 days later. And when we do a hearing test, this is the red line here, the animal is deaf. This is an auditory brainstem response measurement. And on the bottom in black here is the normal ear. This is a normal hearing chicken. What happens over time, though, is that within 4 to 5 weeks, the animal completely recovers. And the test is here. This is the measurement. And on the right, you see the cross sections through the chicken cochlea, and you see the occurrence of new hair cells. And you actually see the dead hair cell debris still on top floating because this is the scala media. This is a fluid-filled space, and there's no immune system that can actually clean up all of the cellular debris. But it's a nice indication that this was originally damaged and has completely recovered. So what I showed you in the last slides is data from my lab. But we did not discover all of this. We simply developed a reliable method to kill all hair cells in one ear without affecting the other ear. And my work and the work of my laboratories group is building on this technological accomplishment. We are standing on the shoulders of the people that I mentioned before and many others who have worked out a lot of the technologies that we are now utilizing in my lab. For example, we can provide a marker that labels dividing cells. So what you see now on the top is the same view again of the control cochlea. And on the upper right, you see a close-up of the supporting cell nuclei. And they're all green, as they are because they're labeled with this antibody, with this label that we use to recognize the cells. On the bottom, you see what happens 48 hours, 2 days after we kill all the hair cells. These nuclei are also positive for a marker that is called EDU, and that marker labels the genetic material of cells that are going into cell division. So this allows us to really show which cells are starting to divide. So this is nothing new. Has been shown in 1988, right? But it's in technical lore. Because we can really see the cells very clearly, we can now look every single day after hair cell loss and during the recovery, how the hair cells, the new hair cells look. And they start to develop their apical specialization, which is the top of the hair cell, where the mechanosensitive organelle, the mechanosensitive structure, is localized, which is called the hair bundle. And a normal hair bundle looks like what you see on the left. And you see there are some scales here because we measure everything. I'm not showing all the measurements. I'm just showing you the pretty pictures here. And what you see is that over time, after 7 days, we see a tiny hair bundle coming up. After 9 days, it looks almost normal. And after 11 days, we cannot distinguish the hair bundle anymore with respect to its length and its width from the original hair bundle. So it takes about 11 days until this top of the hair cell is being completely rebuilt. And these are new cells. When we look at the bottom end of a hair cell connects to the nerve. It's like the microphone. At the top, you have this bulb. That's the mechanosensitive area. And on the bottom, you have a connection to the cable. And this connection is called a synapse. And on the left, you see these synapses labeled with green dots. So this is a normal hair cell, and you see the synapses down here. Over time, these synapses reappear, and the connection with the nerve is being reestablished. And it takes about 11 to 14 days for that. And if you now think back to what I showed you 3-4 slides ago, when we looked with the hearing threshold recovery, the hearing threshold was measurably better after about 11 to 14 days. So this really correlates extremely well with our physiological measurements. What we also found, what others suggested, but what we think is now the principal mechanism of hair cell regeneration in the chicken cochlea, is that it's a cell division based mechanism. So as the hair cells die, shown here, the remaining cells need to close up the epithelium. It's like a wound that needs to be closed up because otherwise material and liquids will ooze through, up and down through this wound. It needs to close up very quickly. And there's a mechanism for that we are interested in. And very quickly, if one hair cell dies in this area, one supporting cell, not two, just one, is being selected. And this supporting cell responds by going back into the cell cycle, meaning it starts dividing, and it divides into a replacement hair cell and a replacement supporting cell. And this is happening after 4 to 11 days. So this hair cell is juvenile. It is not mature. It doesn't function at this point. But then after about 11 to 14 days, it functions. So this is the morphological characterization of the recovery. And the reason we were able to utilize single cell technology is because we had worked out this time course, and it was an extremely reliable time course. And that allowed us to design an experiment like this, where we induce hair cell death, and P7 means post-hatch day 7. So these are fluffy yellow chickens, 7 days old. We induce hair cell death in 1 year, and the hair cells die between 12 hours post-sizomycin treatment, post-hair cell loss, and 20 hours. So this is the time when the hair cells die. And the supporting cells start to divide after 38 hours. So we picked these time points, and then we isolated individual cells from these time points, from these animals. And we subjected them to genetic profiling. This is complex. I will explain it. Every single dot here represents a single cell from this experiment. And the 4 different colors represent the computational calculations that that the computer provided for grouping these different cells into groups of cells that are more similar. So we now have 4 groups, S1, S2, S3, and S4, representing 812 supporting cells. And the grouping is based on 16,302 genes. So the different levels of gene activation in these cells was used to group them. But we also have the original information, namely when we isolated the cells, where do these cells come from? 12 hours, 16 hours, 20 hours after damage induction, for example. So combining these 2 data allowed us to identify group number 3, S3. And S3 is on the bottom here. It consists of 95% or 97% of cells that came only from 12 hours, 16 hours, and 20 hours. So this group of cells is representing supporting cells during the time when the hair cells die. And it also tells us that there is activation of certain genes in this cell group. And having the data on all the genes, we can now use the computer to look up what kind of genes are being activated. So using computational approaches, we were able to determine a novel signaling pathway that we ultimately tested experimentally. I will walk you through the schematic of this. So when the hair cells die, they release certain enzymes. And we think that one of these enzymes is a protease. That means it's an enzyme that cuts off a piece of another protein, very specifically. And this cutting off of one end of this protein called F2RL1, which is a receptor, will activate this receptor. Subsequently, this activation signals to the cell's nucleus, where it activates a gene. And that gene is called HBEGF. HBEGF is then being turned into a protein, the protein HBEGF, that is being presented on the cell membrane. It's anchored to the cell membrane, showing here with this little lollipop. An additional enzyme that is also floating in the surrounding of the dying hair cell will trigger a cleavage of this HBEGF and will release this little ball that binds to a specific receptor. This is called shedding. And it leads to the autoactivation of a supporting cell. This then triggers a signaling cascade inside the cell that ultimately leads to the activation of additional genes. And these genes, when we look at them, they are all involved in cell proliferation, cell division, and regeneration. So this pathway is sort of the smoking gun that allows us to now probe it and to see, well, how essential is it actually? So how can we know all of this? Because I told you I have a couple of balls and a couple of illustrations. I will show you an example. These components of this pathway all have already pharmacological reagents that can be used to inhibit their activity. There's a huge catalog of drugs that are being developed, that are being catalogized, and then they are available. So we can just say, well, okay, this is this gene, it encodes this protein. This protein is a receptor. Maybe there's an antagonist, an antagonistic reagent that can block the receptor. So we found one for F2RL1. We found one for the enzyme that cleaves HBEGF. We found one that inhibits EGF receptor. We found one that inhibits the signaling molecule, and so forth. So when we added these molecules, we see the following. It says, I'm not walking you through the whole paper, it's a lot of data, but I walked you through one experiment. This is the normal situation. 48 hours post hair cell death, we see this incorporation or this labeling of supporting cells with EDU. Here's the Here's the picture, and this is when we count it. In the control situation, there's nothing. There's no cell division happening in the chicken inner ear unless there's damage. Now we have this damage. And we also know that in response to this, we get HBEGF messenger RNA upregulated. We know supporting cells divide, and we know that new hair cells appear after a week. When we now block F2RL1 with this antagonist, we see, shown here in that inset, there's no more EDU labeling, and the EDU labeling is significantly reduced, massively reduced. We do no longer see HBEGF mRNA upregulation. We do no longer see supporting cell division, and we do no longer see hair cell regeneration. This means that this protein and this gene that encodes this protein is essential for the hair cell regeneration in the chicken. I want to stop here with the chicken work. We haven't solved everything there. We know a pathway, but we know that there are different elements of this pathway that we still try to understand. We also have knowledge that there's a second pathway that is happening at the same time that seems to be also essential. So there's a lot more work that needs to be done before we have a full understanding. I imagine that we have seen the tip of the iceberg, but we have seen something that is different from what people have seen before, because we went to the core of the problem. We analyzed the changes in gene expression at the perfect time to identify these novel pathways. So what is the next step? Obviously, we need to go to the mouse right now. Cannot go directly to the human. I cannot imagine that we can just skip an animal model, because we need to make sure that it works, right, in a mammal that naturally does not regenerate. So what we are currently doing is to establish the same hair cell ablation method in the adult mouse. And I put a check mark on this because we have done this. We published the paper last year, and we can get to the point where we can ablate all hair cells unilaterally, meaning on one side of the mouse, adult mouse inner ear, and that's shown here on the left. The bright stuff here are some of the supporting cells that are really full of a protein called filamentous actin that makes them really rigid. But the magenta inner and outer hair cells are gone. This allowed us to determine the time course. And interestingly, the time course of hair cell death in the mouse is slightly different from the chicken. The outer hair cells die within 14 hours. So in the chicken, it takes about 20 to 24 hours. So the outer hair cells die quicker. And the inner hair cells, they survive until day 3, and then they die. So we now have a time course, and we know when to look. At this point, our current focus is to conduct molecular analyses, mainly doing the single cell studies that we have done in the chicken, in the mouse. We know that there's no regeneration in the mouse, right? So the question is, do the cells respond in some way that we can make sense of, that allows us to say, well, they recognize something, but it's not enough to push them back into the cell cycle or push them into cell division and into regenerative, into a regenerative pathway. We can also utilize certain tools, and I will get to this in a second, that have been recently developed for inner ear gene therapy to deliver certain genes. So for example, can we deliver the genes that we have identified in the chicken that push cells into regeneration? Can we deliver the same genes, the equivalent genes from the mouse genome into the mouse? Or can we do a combination of drug treatments? I showed you that we played a lot with antagonists, inhibitors, but we can use the activators of these different proteins that we have identified in the same manner. So this is now allowing us to do very targeted experiments in the mouse. And of course, can this be developed in a future human therapy? We are a long way to get to this point, but we are on it, right? So I want to step back here, and I want to talk about what are we trying to achieve? What are the expectations? So thinking about cures for hearing loss, the cochlear implant is there. It's available. It has come a long way. I mean, the initial cochlear implants were really primitive and a single electrode, right? And now we have multi-electrodes. We have very powerful computers connected to them. It's like the evolution of the telephone. And we currently have a telephone that is really cool, but who knows what we have in 5 years. The cochlear implant is continuously developing and evolving. So this is actually a great challenge to develop a cure that can ultimately compete with a cochlear implant in some aspect for some patients. And that brings me to the question is it possible to have a universal cure? And if you think about the different causes for hearing loss, aging, genetic, noise-induced hearing loss, drug-induced hearing loss, the differences among people, it becomes clear that it's probably not going to happen to have a universal cure. And aging is one example. Cells do age. Cells age, our skin cells age, and we age, and the cells accumulate damage, and their genetic material changes. Cell housekeeping can become messy. The mitochondria inside the cells don't work as good anymore as before. And this is what happens when we simply age. And cellular aging does not only affect hair cells and the organ of Corti, but it affects also the adjacent structures. In fact, the whole body, right? So it's a complex problem. If you fix the hair cell, but another structure in the ear is not working correctly, you will not be able to fully restore the function. You may be helping it along. You make it work better, and you may gain 10 years, but a cure, a rejuvenation is a long way out. This is not Star Trek, unfortunately. But some causes for hearing loss affect individual cell groups only, and cell groups where only a specific gene is active. And this was utilized recently in the well-documented and in this gene therapy trial from Lilly and Regeneron and a couple of companies in China. And there's a company in Europe that does a similar thing, which is the gene therapy for the otolaryngology gene. And I think this is remarkable. It allows profoundly deaf children to hear without a cochlear implant. And I want to explain very quickly what the issue is with this OTOF gene and how it was fixed. So people with a mutation in the OTOF gene cannot hear because information between the hair cell, shown here in red, and the nerve, shown here in yellow, is not transmitted properly. So that everything is intact, just this transmission does not happen. There's a part that is missing. And the otofurlin gene therapy, OTOF gene therapy, provided that genetic material to hair cells. And adding the intact gene now makes this transfer of information possible. And the children hear again. It's just remarkable. This is a single gene. And if you are affected by genetically caused hearing loss, and you do not know what gene defect you have, the chances are 1 to 3% that you have a mutation in the OTOF gene. Depending on your region and your ethnicity, this number could be higher. It has been estimated that potentially about 20,000 patients can qualify for this gene therapy. But worldwide, we have 450 million patients who have debilitating hearing loss, making the market rather small for this very specific therapy. But it's still very important because what it has shown is that we have replacement parts that work. And the tools to deliver these replacement parts are also developed. So these tools, I steal this from the car because my car tends to break down all the time. So you need to fix it, right? And having the tools to get this relay in is going to be very important because you have to really deliver it deep into the engine, right? Deep into the car. You could imagine that you can now use a different gene, and there are some genes that can be fixed. There are other genes that cannot be fixed for certain reasons. But you can also imagine, well, can this be used for regeneration? And I mentioned before, we are trying to repurpose these tools for investigating regeneration in the mouse. But it's challenging because we are probably not dealing with a single gene. We are dealing with moving cells into cell division, which is inherently challenging because we don't want to cause a cancer. We don't want to cause a car wreck. So efficiency and safety are the main challenges that we really need to work out. So I hope I can leave you with the idea that the cochlea is a complex machine. You knew already that it's a complex machine. There are different parts and different areas that can be fixed, but there will not be a one size fits all fix. But thinking about where medicine is going and the current talk about precision medicine, where you really need to understand what is broken in order to fix it, is going to be really important. You don't want to have a therapy and want to have the danger of going through a surgical kind of intervention without knowing whether it will work. You want to know that a treatment will work. With that, before I take questions, I have two things. One is, without HHF funding, we would not have been able to do this work. We started in 2015 around that. It took 7 years until we began to get funded from the NIH. And it supported the careers of several of my people, which now move on to start their own labs. So we propagated it. So we expand the whole group of people that work on this problem. I sort of calculated on a napkin that for every dollar we got from HHF, we were able to leverage $8 from other sources, NIH, other foundations. This is remarkable. And we're now talking to biotech experts to really try to move this forward into treatments. Currently, I have 3 instructors named here, 3 postdocs named here, 3 graduate students, and 1 visiting medical student who work on these problems. Many other people have contributed. If you're interested in what we are doing, we have a website. If there's something significant, we will post it on the website, and you can always go there. I generally appreciate the work conducted by my colleagues that really pointed us in the direction that we are currently taking. Many are connected to the HHF, and all my colleagues in the Hearing Restoration Project, which is a very collegial kind of group of people where we hash out new ideas all the time. This is the funding where it comes from over the years. This is not where we currently receive funding. We only have 3 funding sources right now, but a lot of foundations have supported this. But HHF stuck out. It massively helped us to put this research on a consistent trajectory that allowed us to start work that was not fundable with conventional ways. And without the HHF, we wouldn't be where we are right now. It's long term, and thank you all for listening.
Well, Stefan, on behalf of our audience, which have left many comments, they think fantastic work, simply amazing work, and so forth and so on. And so I too join the chorus congratulating you and your team for the amazing work. I thank you for talking about how Hearing Health Foundation's helped. And of course, and we take pride in your accomplishments, of course. We loved your artwork with Christine Graylap, amazing artist, really beautiful illustrated work. There's a question, you showed the regeneration after following a specific agent. Does it matter if you use different injurious agents to the check cochlea? Is it the same processes or different? Can you comment about that?
That's a good question. We know personally, we haven't done this. We haven't exposed chickens to sounds, but we are starting with this in the next month. I presume it's similar, but sound exposure is more messy. With the sound exposure, you're not ablating all the hair cells throughout the chicken cochlea. You're affecting the high frequency cells more, and you will have this gradient of damage that allows your analysis to be a little bit more tricky. We initially chose this scizomycin because it was really a hammer. It killed all the hair cells, and it allowed us to have a very clean baseline. And in future years, we need to catch up with all these other modalities that cause hearing loss, including noise-induced hearing loss. And then thinking I may reveal that we recently received a grant from SpaceX, which studies the impact of noise on the bird population near their launch site at Vandenberg Air Force Base. And we are hoping to actually measure how rocket launch noise affects the hearing of chickens, which substitute for the endangered species that are nesting around that launch site. And in addition to that, we will perform laboratory controlled experiments to really measure the impact of the intensity levels of rocket launch and sonic booms on avian hearing. And hopefully these animals will regenerate, because if they don't, it will mean problems for.
There are a few interesting questions about this, which is, even though it's just a unilateral hearing loss in the chick that you're inducing, are there behavioral changes you see in the chick with the hearing loss? And number two, when you regenerate the hair cells, is there any way to assess the quality of hearing? Is it the same as before? Can you take that question any way you want to and give us some insights?
Yeah, in chickens, it's hard. The second part of the question. The first part, we do not cause hearing loss. With the hearing loss comes also a vestibular defect. We eliminate also the hair cells in the vestibular system. So the chickens, during the first couple of days after the surgery, don't do really well. They have their head on the side, it's tilted, but they recover from that after a couple of days. It's also because they have vision, right? If we do this unilaterally, the chickens are relatively normal. I think if we eliminate the hair cells in both ears, the chickens will have a horrible couple of days. They will not be able to feed, so we actually don't do this because you have to hand feed every single animal, and the unilateral approach, I think, is better for us. With respect to the quality of hearing, that's a really good question. We cannot talk to the chickens. We can only measure a nerve signal, right? And that is restored. There have been studies to eliminate hair cells in songbirds. And songbirds have the ability to learn a new song, right? And when you eliminate hair cells in a songbird and you present a songbird that has recovered from that with a new song, these birds are capable of learning a melody. And that tells me that there is that the hearing quality and the connections in the central nervous system are being properly restored. That's the only measure that I can currently think in birds that allows us to make a statement about whether their high fidelity of the hearing is actually working.
That's a great insight, Stefan, about the songbirds and their ability to maintain that. It might speak to their qualities. So that's a great analogy there. Now, you showed data about how the synapses come back. Were the synapses lost in the first place? Do you lose the synapses after you lose the hair cells? And also, if we regenerate hair cells down the road, will the synapses come in humans as well? So take that question broadly and talk a little bit about synapse regeneration versus hair cell regeneration, which occurs first, which occurs after. Is it which one is the tail, and so on?
Good question. I mean, we looked at this with a very naive kind of initial view, and then so we realized the hair cells are gone. So the nerve that connects, the hair cell is here, the nerve comes in here, connects to the hair cell. The hair cell makes poof and is gone. This nerve will retract about about 5 lengths of the individual hair cell to the bottom of the cochlea, and it will just sit there. And when a new hair cell forms, that new hair cell, and this is a perfect example from about the difference between development and regeneration. During development, when the ear forms, the hair cells secrete a neurotrophic factor, that one of these factors I mentioned during my talk, called BDNF. And that neurotrophic factor attracts the innervation to the hair cell. So the nerve grows toward the hair cell. But this nerve at this time during development is not properly connected in the brain to the right frequency. So these cables haven't been really sorted out. This is high frequency, this is mid frequency, this is low frequency. So during development, the hair cell utilizes this neurotrophin. If you provide a neurotrophin to a regenerating cochlea, and I haven't done this experiment in vivo, but if you do this in a culture dish, it leads to a wild nerve growth called sprouting. So this nerve will go in all directions. So if the hair cell would be utilizing this neuroattractant during regeneration, the result would be a mess. So the cable that has retracted to connect this, let's say 2000 hertz hair cell to the 2000 hertz mapping into the brain may move in a different direction, and it may move to a 1700 hertz hair cell. So your hearing will not be restored with high fidelity. So what instead happens, the new hair cell is formed with a process that reaches straight down to the place where the nerve has retracted to in a location-specific manner, and it makes contact with the nerve on the bottom of the cochlea. So this preserves the frequency specificity of the regenerated hair cell with the nervous system. So the birds have worked out this regeneration completely fine, and and that's probably why the songbirds have pretty much perfect hearing again. At least that's my interpretation. Whether this is possible in humans, well, we have to see whether a newly regenerated hair cell is capable of growing this process that reaches down to the nerve. There are two ideas that make me feel positive about this. One is that the nerve retracts in the mammalian ear also very slowly, and it doesn't sprout a lot. It doesn't change its location a lot. That's also why cochlear implants are very stable. I mean, you put them into babies, and I don't know what you probably know, the oldest cochlear implant person who has received a cochlear implant as a baby, and how hearing is hopefully permanent in these patients. But I think that hearing is pretty stable, and once it's fully established. So these nerve fibers will not go in wild direction in a mammal either. We know that if you generate a new hair cell in the mouse and in the rat, that it attracts nerve fibers, and the nerve fibers make connections to these new hair cells. If we make a new hair cell in a more natural way, like the chickens make new hair cells, these hair cells may look different than the reprogrammed hair cells that are showing up in papers over the last 20 years, right? These are cells that just are being forced to take on a hair cell fate, but they are not undergoing this regenerative rebirth that supporting cell-derived and division-derived hair cell experiences. So we have to recreate this in the mouse before we can answer the question. I'm of good hope. And if you go to other organisms like zebrafish, a similar phenomenon has been reported there in regenerating hair cells. So it seems to be a general mechanism through different organisms. And we know that evolutionary, we are all related. So this program is maybe dormant in mammals as well.
So I'm going to apologize to our attendees who have many good questions, and we're getting down to pretty much our last question right now. And broadly speaking, how far are we out from a cure for hearing loss? And I think, and I asked this question. One of the attendees asked it. And
Mm.
I want you to answer that in any way you can, and give us what this future looks like for us,
Mm.
and how far is that future?
I think we're already there, but we don't realize it. I'm just going back to that example that I had with the telephone. The original communication happened with the pony trail, right? And then we had Morse code, and that allowed us to send messages. So we are at these early stages, and we have now the ability to cure genetic hearing loss in a very specific subgroup of patients. Check. So we made the first step. And I think some of these 450 million patients, if you break this up in a pie chart, it will have a lot of pieces. And some pieces of this pie will be large, and some pieces will be relatively small. And these pieces represent patient groups. And the sliver of pie that is represented by otofurlin patients, they will benefit. For them, we already have a cure. The cure, I don't know how permanent it is. Same question. If a child receives a cochlear implant, will that child, when it is 80, still benefit from the cochlear implant? I don't know that, right? So, but it already happens. So now the question is, how fast will this pie open up? And how many of these pieces of pie can we accommodate in the future years? And I think it will be a couple. It won't be the whole pie in the next 10 years, but it will be a couple of more pieces in the next 10 years. And hopefully this will accelerate everything. Because I mean, as soon as the biotech companies that have invented this gene therapy, they got gobbled up by Lilly and Regeneron for hundreds of millions of dollars, right? So these large pharmaceutical companies see that there is a potential market there, and the market is listening. I mean, we all, and more people are the market. My hearing is not as good as it was when I was 20, and so there is an opportunity for business, and I hope that this will lead to more efficient and more safe and safe treatments, and that more money will come into our field through biotech and through pharmaceutical companies. Biotech, the history of biotech in the inner ear is not a good one. I mean, there are lots of failures, but the complexity of the problem is really high. And as I said, this pie is really a complex pie. We have so many different pieces and so many different patients who are affected because of different causes. So recognizing what is wrong with these patients, finding the right diagnostics to properly say, this is the problem. Like with OTOF, we knew exactly what the problem is. It took 20 years to identify this problem. The first papers go out 20 years. And now coming up with a fix took 20 years. So just project this time frame. It will take time to get to this point. I'm inherently an optimistic person, and sometimes I have a tendency to be a little bit pessimistic, but ultimately we are already en route. We are getting there. If a specific patient now has the question, when will it benefit me? That patient needs to know what the cause of his or her hearing loss is. So they need to talk to their otologist. The question is also, if they are at a certain age, does it actually make sense for them to consider anything that is purely experimental, whereas the gold standard, if you're really profoundly deaf and you have hearing loss, the gold standard is still the cochlear implant. And if I would be affected at my age right now, I wouldn't hesitate. I would actually get a cochlear implant, still work on treatments, try to help people in the future. I try to be realistic here, but we are on our way.
I think you're absolutely right about the fast pace. I mean, it was just 30 years ago that my lab in San Francisco showed AAV as the vector in gene therapy, and
Right.
we already have a therapy now, and we're looking for the same accelerated pathway from your work to human regeneration of hair cells. Your conversation really made me hungry with lollipops and pie for food, but also for new knowledge and direction. So, Stefan, really just an amazing talk. Thank you so much. I also want to take a moment to thank our attendees. We're so grateful to you, our community, for your support of our Emerging Research Grants Program, ERG, as well as the Hearing Restoration Project, which Dr. Heller is a member of. Now, you remember you can donate to our efforts to advance better treatments and cures for hearing and balance conditions at HHF, Hearing Health Foundation, hhf.org/donate. And thank you, and please enjoy the rest of the day.